Introduction to inflammation. Inflammation is a biological response to harmful stimuli such as pathogens, damaged cells, or irritants. When harmful agents enter our body, inflammation serves as a protective response. The body sends immune cells to the affected area to neutralize the threat. Inflammation is a protective mechanism with two main purposes. First, it removes harmful stimuli like pathogens or damaged cells. Second, it initiates the healing process to restore tissue function. However, inflammation has a dual nature. It can be both beneficial and harmful. Acute inflammation is beneficial, protecting against infection, removing damaged tissue, and promoting healing. Chronic inflammation, however, can be harmful, causing persistent tissue damage and being associated with various diseases. This overview is based on academic research from Intech Open Publishers and work by Anne C.F. Nunez from UC Irvine. The cardinal signs of inflammation have been recognized since ancient times. There are 5 classical signs of inflammation: redness or rubor, heat or calor, swelling known as tumor, pain called dolor, and loss of function or functiolesa. Let's start by examining normal tissue with blood vessels, cells, and nerve endings. When tissue becomes inflamed, it undergoes several dramatic changes. Redness, or rubor, is caused by increased blood flow to the inflamed area. Blood vessels dilate, allowing more blood to reach the affected tissues. Heat, or calor, occurs as increased blood flow brings more warmth to the inflamed area. Additionally, inflammatory mediators increase metabolic activity in the tissue, generating more heat. Swelling, or tumor, occurs when fluid leaks from dilated and more permeable blood vessels into surrounding tissues, creating edema. Inflammatory cells also migrate to the area, contributing to the swelling. Pain, or dolor, results from stimulation of nerve endings by inflammatory mediators such as prostaglandins and bradykinin. Swelling also creates pressure on nerve endings. Pain helps limit movement of the injured area to promote healing. Loss of function, or functiolesa, results from a combination of pain, swelling, and the physiological changes in the tissue. Movement becomes difficult or impossible. This limitation can be protective, preventing further injury and allowing healing to occur. To summarize, the 5 cardinal signs of inflammation are redness due to increased blood flow, heat from increased temperature, swelling from fluid accumulation, pain from nerve sensitization, and loss of function, which limits movement. These signs are the body's response to injury or infection. Inflammation can be triggered by various threats to the body. These include pathogens like bacteria, viruses, and fungi. Physical trauma such as injuries, burns, or frostbite can also initiate an inflammatory response. Chemical irritants, including acids, toxins, and allergens, are another common cause of inflammation. Finally, autoimmune responses occur when the body mistakenly recognizes its own tissues as foreign, triggering inflammation. Immune cells detect these threats using specialized pattern recognition receptors, or PRRs, located on their surfaces and within the cytoplasm. The main types of PRRs include toll-like receptors, nod-like receptors, and rig-like receptors, each specialized for detecting different threats. PRRs recognize 2 main types of molecular patterns, PAMPs and DAMPs. PAMPs are pathogen-associated molecular patterns, unique molecules found on pathogens but not in the host. These include bacterial cell wall components, viral nucleic acids, and fungal glycoproteins. DAMPs are damage-associated molecular patterns, molecules released from the host's own damaged cells. These include cellular debris, heat shock proteins, and ATP released during tissue injury. When a pattern recognition receptor binds to its target molecule, either a PAMP from a pathogen or a DAMP from damaged cells, it initiates a signaling cascade within the immune cell. This recognition is crucial for the body to identify threats and mount an appropriate inflammatory response. Without this recognition system, the body would be unable to respond effectively to infections and injuries. The inflammatory response involves several specialized immune cells that coordinate to defend the body. Neutrophils are the first responders, arriving at the site of injury or infection within minutes. Macrophages function as the cleanup crew, engulfing pathogens and cellular debris. Mast cells act as alarm signalers, releasing histamine and other inflammatory mediators. Lymphocytes, part of the adaptive immune system, provide more targeted immune responses. Neutrophils are critical first responders in the inflammatory process. When inflammation begins, chemical signals called cytokines trigger endothelial cells in blood vessels to express adhesion molecules. Neutrophils in the bloodstream are attracted to these signals and begin to roll along the vessel wall. The neutrophil then firmly attaches to the vessel wall and begins to squeeze through a process called diapedesis. Once in the tissue, neutrophils migrate toward the site of infection or damage, guided by chemical attractants. Macrophages arrive at the inflammation site after neutrophils. They perform essential functions, including phagocytosis of pathogens and clearing cellular debris. Macrophages engulf pathogens through their cell membrane, forming a phagosome that contains the pathogen. Inside the macrophage, the pathogen is broken down by enzymes in a process called phagolysosome fusion. Macrophages also clear cellular debris from damaged tissues and dead neutrophils, helping to resolve inflammation. Mast cells and lymphocytes also play important roles in the inflammatory response. Mast cells are tissue-resident sentinels that detect tissue damage and pathogens. When triggered, mast cells rapidly release preformed granules containing inflammatory mediators. On the other side, lymphocytes are part of the adaptive immune system and include T cells and B cells. The inflammatory response involves a coordinated sequence of cellular activity. Resident mast cells first detect damage and release alarm signals. This is followed by the rapid arrival of neutrophils. Macrophages arrive within hours, phagocytosing pathogens and clearing debris, including dying neutrophils. Finally, lymphocytes provide specific adaptive responses, arriving days after initial inflammation to target specific pathogens. To summarize, each cell type plays a specific role in the inflammatory response. Resolution of inflammation is not merely the absence of inflammatory signals, but rather an active, highly coordinated biological process. Specialized pro-resolving mediators, or SPMs, are key molecules that initiate and orchestrate the resolution process. These include resolvins, lipoxins, protectins, and maraicins, which are derived from omega-3 fatty acids. The first step in resolving inflammation is the cessation of neutrophil recruitment. SPMs block neutrophil chemotaxis by inhibiting pro-inflammatory signals. The second process is programmed cell death, or apoptosis, of inflammatory cells. SPMs promote neutrophil apoptosis rather than necrosis, preventing the release of harmful cell contents. The third process is the clearance of apoptotic cells and debris by macrophages, known as efferocytosis. SPMs enhance macrophage phagocytic capacity and promote anti-inflammatory phenotypes. The final process is tissue repair and the return to homeostasis. SPMs stimulate tissue-resident cells to promote repair, while macrophages shift to a pro-resolving phenotype that supports tissue healing. It's important to emphasize that resolution of inflammation is not simply the passive decay of inflammatory signals, but rather an active, coordinated molecular program that restores tissue homeostasis. Understanding inflammation has profound clinical implications in modern medicine. Inflammation biomarkers help clinicians diagnose diseases, assess treatment effectiveness, predict disease progression, and stratify patients based on risk. Various treatment strategies for inflammatory diseases have evolved based on our understanding of inflammation. Traditional anti-inflammatory drugs like NSAIDs and corticosteroids broadly suppress inflammatory mediators. Modern biologics like anti-TNF antibodies and IL-6 inhibitors target specific inflammatory pathways. Newer approaches promote active resolution of inflammation using specialized pro-resolving mediators and antifibrotic agents. Current research in inflammation biology is advancing in several exciting directions. The microbiome has emerged as a crucial factor influencing inflammatory processes, particularly the gut inflammation connection. Targeted biologics allow for precision molecular targeting, blocking specific inflammatory pathways with minimal side effects. Personalized medicine approaches tailor treatments to individual patients based on their unique inflammatory profiles. The field of inflammation research continues to evolve as our understanding deepens. Past research focused on understanding basic inflammatory mechanisms. Current research explores detailed molecular targets and signaling pathways. Future directions include systems biology approaches and artificial intelligence integration for complex data analysis. This overview is part of a larger body of translational studies that continues to evolve as researchers gain deeper insights. As our understanding of inflammation deepens, it continues to drive innovative approaches to disease management and treatment.